Well, good morning, folks. It's me, your old pal, Dr. Harry, back again with another class. So, this morning, our goal is to discuss the autism spectrum disorders. These are, well, we're returning to, I guess, the world of the more kind of complicated polygenic conditions that characterize, frankly, most of mental health and various aspects of psychology. We had a nice time looking at the sort of monogenic conditions like Huntington's and Fragile X, but we're back into the world of the complex. Now, there is an interesting connection between this lecture and the previous two that I will get to, which is that, as I will discuss, emerging evidence is pointing to a role for repeat expansion in the genesis of ASD. So it may be actually the case that there is more connection between the previous conditions and this one than you might have guessed. Anyway, let's jump into it, my friends, and let's start learning about ASD. So here's the title slide. The topic is Autism Spectrum Disorders. Hold on a second here. Let me just adjust my slideshow thing. Okay, there we go. So the autism spectrum is a somewhat novel interpretation of the syndrome known as autism up until the DSM-5, which I believe is still the most recent version of the Diagnostic and Statistical Manual for Mental Illness. Individuals who were on the extreme end of the autism spectrum were diagnosed with autistic disorder, whereas those with higher levels of function were described as having Asperger's syndrome. Since the DSM-5, and I think probably since most of you have really been conscious of mental health in your life, that's actually become kind of outdated. The favored terminology now is to just call everything the autism spectrum disorder, or ASD. And then you're given a numerical grade based on the level of functional impairment. So, a grade like 3 would mean that you require a great deal of support in your day-to-day life, whereas a grade level of 1 means you require some support. And of course, if you require no support, it's difficult to really say you have a disorder, isn't it? But of course, that brings up kind of the complicated interaction between disability and society, because the level of support you need isn't only a function of your own capabilities, but it's also a function of the way society is set up so as to enable or to support you to varying degrees. So there is a bit of an interaction there. But anyway, high functioning autism and Asperger's syndrome are now diagnosed as different levels of the autism spectrum. So when we're talking about ASD in the context of genetics and epigenetics, like we'll be doing today, this is kind of the spectrum we'll be working with. And I suppose quite typically, researchers, if they agree that there is a spectrum, they can capture a wider span of people, maybe with varying levels of impairment, to get more data and more numbers in their studies. I suppose this whole thing is a little bit like when they declared that Pluto isn't a planet. It's justified, but it's in a way a little pedantic. You argue so much over the classification of things and bring things in and out of the disorder category, whether they think they don't exist. It sort of amounts to a kind of stamp collecting. And I mean, like the way we speak about disorders, I've brought this up a little bit, but the way we speak of disorders is in the popular mindset or the popular culture is quite separate from the way they're treated clinically. But that's the way language works in general. The way we talk in our kind of day-to-day lives doesn't have a lot of correspondence to the way the actual science of things works. So for example, we talk about sunrises, even though we've known for many, many centuries that what's happening is not that the sun is rising over the horizon, but rather our movement, or the rotation of the Earth relative to the sun, brings our part of the Earth in view of the sun, and that's what's actually happening. So the sun isn't rising literally, but the term is still there. Anyway, interactions between science and language, I guess. Pre-scientific modes of speaking are probably permanently fixed in our language. And really, I guess if you think about it, even post-scientific terms are also kind of fixed in our language. We talk about, Freudian slips, for example, even though Freud's theories aren't exactly favored these days, some things just kind of stick in culture, and they reflect outdated or perhaps pre-scientific explanations of things, but they evoke such a powerful image in our minds, or they've become so fixed in our language that we use them anyway, maybe knowing in the back of our mind that they're only analogies or only sort of like poetic descriptions of things. So ASD, these disorders typically appear in early childhood. Of course, they're considered sort of neurodevelopmental disorders, usually recognized around the second year of life, once the child becomes a bit more social. Once the child is starting to use language, you start to be able to notice deficits compared to average children. Although child development at this age is quite variable, it's really surprising how similar kids are in their abilities by the time they enter school, relative to how different they can be at age 2. You'll see 2-year-olds walking around counting and speaking pretty fluently. You'll see others who are not yet verbal and have rather limited capacities, and they all end up to be pretty much the same by the time they get to school. So sort of funny how that can be. But anyway, the coincidence of social development and the appearance of ASD is no coincidence. Parents will often report a fairly normal pattern of development for the first year or so, and then they'll report a decline in the following years. And this is, in a sense, it's a decline relative to the expected gain in capacity that you would see with a child. They would display some beginning stages of social behavior, eye contact, and noticing the appropriate cues in society, and maybe using words here and there, and then they would start to sort of regress. And this timing is unfortunate in the sense that it happens at the same time children receive a lot of their immunizations. The coincidence of those two events has led to a lot of sort of conspiracies about vaccines and so forth. It's difficult to kind of, the timing of things is always very difficult for people, because really, you only have one child. If two things happen in the child's life right next to each other, and at the same time you're noticing a problem with the child or a decline in function, it's very difficult to not make a connection. And science is filled with enough ambiguity, and there's enough complexity to science that you can kind of look at the literature a lot of different ways and make up a lot of, come to a lot of conclusions about what's really going on based on your reading of the literature, because nobody has the insight to understand everything perfectly well. They sort of make interpretations based on their own life experience and what they can interpret from the literature. So that's really not a great thing. But anyway, the autism spectrum disorders are characterized by 3 major areas of difficulty. Those would include impaired communication, impaired social interactions, as well as restricted behaviors, interests, and activities. I suppose you probably, I don't need to really go over this. I'm sure by year 3 of your neuroeducation, you're more than familiar with ASD. So ASD affects about 1 to 1.5% of the population, although it's sort of, it's not exactly, we're not exactly certain, how many people truly have this, because the diagnostic criteria are sort of defined relative to what's expected out of society, and people become more and more aware of the ASD phenotype, and thus the rate of diagnosis can go up. Other developmental disorders become less common at the same time. So, but it affects around 1 to 1.5% of the population, depending on exactly where you draw the cutoff lines, and that's a fairly large number of people. That's pretty similar to the number of people who have schizophrenia. Autism has a clear genetic component, and many decades of observation have shown that autism spectrum traits seem to run in families. This is kind of responsible for many of the observations made, showing, for example, that rates of ASD are higher in, Silicon Valley and places where a lot of tech-focused people go. The theory being that perhaps ASD, the genes that cause ASD in lower doses, or when you have fewer of these particular alleles, they lead you to become more analytical and more, more analytically minded, and thus more appropriate to careers like mathematics and engineering, and I guess maybe STEM more broadly. But if 2 people with those same kinds of traits get together, the odds are increased that all of those genes would converge and maybe push their child past the breaking point where they're no longer helpful, but they're actually sort of harming neurodevelopment in some sort of way. That's an attractive theory. When you actually look at the real genetics of it, which I'll be giving you, it doesn't really pan out that way. These kind of observations are really interesting as theories, but they don't really correspond terribly well to the actual genetics of things, unfortunately, because it's a wonderful kind of perspective on things. Looking at twin studies, these suggest that the heritability of autism spectrum disorders range from between 37 to 90%, which is quite a wide range, but again, that depends exactly on what cutoff you draw. Are we talking ASD, or are we talking about sort of full-blown autistic syndrome? Difficult to say. And they're about 4 times more common in males than females, which is another kind of interesting sex difference. It's one of the stronger sex differences you can find, especially for a condition that's so heavily dependent on genetics, or rather that appears to be so heavily dependent on genetics. And most of our data don't really get at the answer to this question. The thing you have to think about when you're looking at sex differences in genetics is that males and females have almost all of the same DNA. You know, if you have, say, a sister or a brother, or, you know, a sibling of the opposite sex, the only real difference genetically that they would have, I mean, they'll be different in many, many ways, but like the only meaningful difference between you and them is the presence or absence of a Y chromosome. You know, they'll have XY or XX. And that's only one chromosome out of 20 plus that humans have. So it's actually like the genetic influence of this is fairly minor. You can't explain sex most of the time, I should say, you can't explain sex differences like this by the sex chromosomes, because the genes that appear to be related to ASD are on all of the autosomes, or they're spread widely through the genome and loaded fairly heavily onto autosomes. And you and your sister or brother, you and your opposite sex sibling, have functionally the same sorts of autosomes. I mean, they'd be different because you inherited them differently, but they would be coming from your parents the same way. So how can you explain sex differences in the manifestation or the expression of a phenotype when the genes that appear to cause it are all on non-sex chromosomes? You have to posit kind of an interaction between the fixed genetics of the disease and the more plastic, hormonally, developmentally driven nature of sex, and imagine somehow that the presence or absence of sex hormones or other kinds of sex-specific features kind of unlocks the genetics, or maybe makes them have a stronger effect in one sex or the other. So we will get into this a little bit in the last couple of lectures where I'll talk about this, but it's something to keep in mind. The sex differences are very difficult to explain by genetics alone. You have to incorporate some kind of developmental understanding of the way the genetics unfold. But anyway, females are overrepresented among low IQ cases. So the people who are the most impaired in ASD diagnoses tend to be females, whereas the higher IQ variants of ASD seem to be more common among males. And this, then you also get into questions of like, well, how is this being diagnosed? Is there just a bias in the field of psychiatry as to how the diagnosis plays out? It's tough to say. So I have a question in the chat. Oh yeah, so Alexis St. Pierre asks, why do you think this large sex difference exists? To be honest, I kind of don't really know. I don't think anyone is really certain why this exists. There are a lot of sex differences in disease manifestation, and most disorders or most diseases that are not like monogenic sex-linked conditions, like Fragile X, for example, most of those the genetic basis of them is spread quite evenly across all of the autosomes. So whether you're born a male or whether you're born a female or whatever, you're going to have the same autosomes, or that doesn't affect what genes or alleles you get on your autosomes. So there is some kind of variable penetrance that arises based on how sex differences develop in the first place. So one difference that isn't quite related to this would be the developmental programming of obesity risk. This is something that I study a bit more closely, so I can have a more insightful answer. But in mice, at least, females tend to be a little bit less vulnerable to this. If you mess with mom's nutrition, that has a bigger effect on the male offspring than it does in the females, typically. And the reason we think this is happening, basically, is because males grow at a faster rate. They need to get bigger in the same amount of time than females. They are affected more negatively by changes in nutrition because their bodies have higher nutritional demands in the first place. So if you limit their nutrition levels, the effect it has on development is like relatively larger. Females develop somewhat more slowly. They reach a smaller terminal size, and so they're able to cope with the ups and downs of nutrient availability a little bit better. It's sort of like if there's a gas shortage, the people who drive, giant pickup trucks, F-350s and Hummers and whatnot, are going to feel affected by it a lot sooner than people who drive, a Toyota Prius or a Honda Civic or something like that. So it's that kind of thing. But every disease or every disorder has a different explanation. It's really quite complicated. And in the literature and in science more broadly, it's only now becoming acknowledged by people that you really have to study things from a sex and gender-centric perspective. You can't pick samples of convenience. You can't do your animal studies in one sex or the other. You really have to be doing everything in both in order to understand the true complexity of what's happening. And that's been very, very lacking in all aspects of medicine and science for many, many years. Funding agencies like the CIHR and the NIH in the US are putting in sort of provisos to try to mitigate this or to try to correct this, but there's a lot that has to be caught up on in order for us to really understand what's happening and why sex differences exist to the extent that they do. So ASD has a genetic component, but the search for variants that cause it seem to be difficult to find, I guess. ASD is apparently very polygenic. It's extremely polygenic, meaning that the specific profile of trait-associated variants is very different between individuals. In other words, ASD is genetic, or it's believed to be genetic, but one case of ASD over in this family could be caused by completely separate genes than ASD in another family. The mutations might converge upon the same neurodevelopmental pathways, and they might produce the same phenotype, but they happen in completely separate places. And that poses a rather large problem for GWAS studies, because one of the assumptions of a GWAS study is that the underlying genetic architecture of a given condition is pretty similar across everybody. That's how you gain enough power. Most people, to take an example, a very large percentage of people with schizophrenia have a certain pattern of mutations in the major histocompatibility complex region of chromosome 6. If only 1% of people who had schizophrenia had that particular mutation, it would be very tough to find by using the GWAS method. You really rely on a fairly high level of consistency among people. Either that or you run giant studies with millions of participants. But that's not possible either because there just aren't enough people to even study to get that kind of detail. So it's a rather complicated thing. GWAS studies presume that all individuals share similar genetic architecture. The largest GWAS study of ASD was published a couple of years ago in 2019 by Grove et al. This is the paper in Nature Genetics. As you can see, these big papers, these big genetic studies have huge numbers of authors. All the way starting from Jacob Grobe. I don't know how this fella got to be the first author. How do you swing that? All the way down to Anders Borglum. He sounds Danish. Anyway, and then you also have people like 23andMe. 23andMe is that website where you can send them your DNA and they'll tell you how likely you are to have like high blood pressure or something. So they have a lot of genetic data and they can contribute to these kinds of studies as well. Psychiatric Genomics Consortium, PGC, we've talked about them before, and many, many others. But anyway, this is just to say that like genetics is a big field. It's kind of like quantum physics, where like when the Hadron Collider or when CERN releases a new study, there's like zillions of authors on the paper because everyone gets together and contributes their own piece to it. But this had 18,000 ASD cases and 27,000 controls. Now, that's a very large number of people, but the number is not even remotely comparable to the number of people we've been able to study for things like height or BMI. The reason for this is that everyone has a height. You can take literally 1 million people. Everyone is going to have a level of height you can measure, and you just plot the genetics against their quantitative trait of height. When it comes to case control studies like this with ASD or schizophrenia, much harder to get that number of people, because how are you going to find 18,000 people with ASD. You'd have to look over the entire country. You'd have to be in contact with hospitals and schools and daycares and nurseries and doctors and all kinds of other people all over the country and get these samples, get detailed data on the person that you're looking at, make sure they're balanced for sex and age and all the rest of it. So it's just very tough to make these kinds of determinations, because if a condition is rare enough, it's just very tough to find enough people. This is a big limitation with discovering the genetics of conditions like this. So here's a Manhattan plot. Follows the typical Manhattan plot rules that we all know and love. So out of this giant study, Grove et al. Found 5 SNPs. 5 genome-wide significant SNPs. They're denoted by these diamond shapes. They're sometimes called index SNPs for reasons that I'll get into, I think. Speaking of which, I forgot that I wanted to make a lecture about neurodegenerative diseases. So one of the lectures, the second obesity lecture, is going to be switched for neurodegenerative disease lecture. So we'll learn about Alzheimer's and whatnot. That reminded me because I was talking about index SNPs in that one as well. But anyhow, they found 5. The index SNP is marked with a diamond. It's the one that they genotype that has the highest association with the disease. And then nearby SNPs that happen to be in linkage typically kind of contribute to, or like, it's kind of built on top of a pile of SNPs that are also quite significant based on the basis of linkage disequilibrium. But anyway, so they found 5 SNPs. They plotted, just for kind of comparison's sake, correlated traits like educational attainment and depression and so forth. They plotted those in gray. So these gray points are not from ASD, but they're from traits that are sort of correlated with ASD in terms of their genetics. And you can see that SNPs that are sort of significant or highly associated with ASD also happen to be associated with other neurodevelopmental phenotypes like educational attainment and schizophrenia. So it kind of points to somewhat of a common underlying architecture of disease in a sense, or of phenotype. But anyway, just consider the fact that they had tens of thousands of subjects, 18,000 cases, and all they got were 5 locations. Think about how limiting that is for a condition or for a phenotype that we believe is highly genetic. You only get 5 SNPs. And SNPs, my friends, they don't tend to contribute a whole lot. These are not ridiculously significant p-values. This is not, the p-values for SNPs that correlate with height, for example, have negative log 10 p's, up in the 20s and 30s, like much more significant. So these are very low effect size SNPs by comparison, and they only found 5 of them. So how can you have a genetic trait where you're seeing the best study you can possibly do is only giving you 5 SNPs to look at? Now, that's the state of the art for GWAS. Now I'm going to take some time to give you some other topics about what you can do with the GWAS data, because I think it's worth, as I was saying, it's worth kind of trying to point out when phenotypes like ASD and educational attainment, or ASD and schizophrenia or whatever, when they seem to have a similar underlying genetic architecture, because it may well be the case that all these kinds of phenotypes impact brain development in similar ways by interacting with similar genes. That would be very fascinating, because I think many people in the world of psychiatry and psychology do suspect that there is sort of an underlying similarity, I guess, between things like schizophrenia and bipolar disorder, for example, that is purely on the basis of their behavioral and psychological phenotypes. But if you can find an underlying genetic similarity as well, that really strengthens your case, because behavior is kind of subjective, but genetics at least are objective. So GWAS studies generally work from the same basic pool of variants. When you decide to conduct a GWAS study, they're done using microarrays, and you would order your microarrays from a company like Illumina, and you basically just order one that genotypes as many SNPs as you can possibly get. They'll cover the whole range of known human SNPs, and at this point they cover a lot of them. They cover virtually all of the main, virtually all of the SNPs that are common enough in the population that you could reasonably detect and link to a disease. But they're all starting from the same place, so you can only have so much to work with. But the good news of this is that it's possible to correlate GWAS results from different traits, because you can take the results from each SNP and compare them across different traits and see if they sort of have a correlation. And what this can show you is that sometimes the genetic architecture of different traits is actually surprisingly similar. So this is a concept called genetic correlation. It's actually a concept that predates genomics. They were able to do this kind of stuff with these old fruit fly studies and whatnot. It's beyond me how anyone had the patience for that, but it is possible now that we have genomics and we can do this all on the computer. It's a lot simpler in many ways. So let's take a look at what GWAS datasets actually look like. Because I think to explain what a genetic correlation is in practice, it's useful to know what data you are like what the data that you're correlating even look like. So raw GWAS data consists of thousands and thousands of rows of genotype calls, maybe millions potentially, depending on how many SNPs you're actually genotyping. So every row is a SNP, and every column essentially would be the genotype call on that variant for a certain person. And so researchers will take this raw data and do a whole bunch of statistical analyses to account for linkage disequilibrium, age, sex, disease status, whatever other variables they wish to control for. But roughly speaking, as I said, what lecture was that? Anyway, there was a lecture where I explained how allele counting worked and how you could count up the number of copies of an allele and kind of correlate that with a trait. I think that might have been the schizophrenia lecture, or maybe the neuroanatomy lecture. But anyway, it's one of those earlier ones. What they're ultimately trying to do is determine the extent to which having a given allele is associated with an increased risk of disease. So for every copy of this allele you get, your odds of being diagnosed, say with ASD, go up by 20%. This is the kind of relationship they're trying to establish, and they're doing this for every single SNP in the entire array. So processed GWAS data, the kind we would actually use in our day-to-day lives, is more like what you would see in just a regular spreadsheet. Each row represents a separate variant or a separate SNP that's been genotyped. And then you have, for each SNP, some kind of statistic that reflects its relationship with the phenotype of interest. If it's a continuous phenotype, typically what you would have, a continuous phenotype would be like height or BMI. Height can go from, 4 feet up to 7 feet or whatever. It's just a continuous number. You have a number that expresses your height. For those kinds of things, you would have a beta value. That's essentially the slope of the regression line connecting the genotype to your quantitative trait. If you have a sort of a dichotomous trait like ASD, positive or negative, do you have ASD or you do not, then typically what you would get is an odds ratio. So for every copy of this allele you have, your odds of having ASD go up by whatever percent. So there's sort of two different ways to do the analysis, but ultimately you have a number that reflects the relationship between genotype at that SNP and your phenotype. So I'll give you like a little example of what GWAS summary data look like. This is, I guess this is kind of for those of you playing along at home who are interested in doing this maybe professionally or doing this as a project for your, as a master's thesis or something like that, or working with these kinds of data. I just wanted to give you like an idea of what it looks like, perhaps to show you that it's not really that scary to actually work with these data once you know kind of some of the software like R and things like that for processing the data. So you get, in a typical example, you would have maybe 13 columns. The first column, so every row is a SNP. Let's just say that right off the bat. The SNP ID is given here. These are these RSIDs that you've seen before. Think back to your first assignment. You could punch any of these ones into your genome browser and it would take you to exactly where that SNP is located in the genome. The chromosome is given in the first column. These are all on chromosome 1. The position on the chromosome is given in the third column. So this is actually sorted kind of badly. It's not sorted in alphabetical, or it's not sorted in numerical order. But anyway, coordinates exist on chromosomes, and you can just point to where the SNP is located on chromosome 1. Then you have the two different alleles that are genotyped. The A1 allele is the reference allele. This is the one that is sort of the more standard common allele for people to have. And then A2 is the alternate allele. This is the other version that you could test. So that the genotyping assay is telling whether you have this or that. These are the two alleles. So you could either have so for example, for this SNP, you could have either a T or a G. For this SNP, you could have either an A or a G. So you could have AA, GG, or AG, basically the 3 genotypes that are possible. Then you get the frequencies of those two alleles in cases and controls. So the A allele, or the A1 allele rather, about 25% of cases have it, and then 25.5% of controls have it. So this allele is a little bit more common in controls. And you can kind of go down the line and see, sometimes the A2 allele is actually the more common one in people. Like, for example, here, like less than 1% of people have the A1 allele. That would mean most people have the A2 allele. But you can kind of go down and look at the frequency, and you can see that, for most of these, cases and controls have the same allele frequency. And if cases and controls have the same allele frequency, it means that that allele probably doesn't have very much to do with whether you're a case or control. If one allele was massively overrepresented in cases, cases 90% of them had this allele and controls maybe only 5% of them had it, that would tell you that allele probably has a lot to do with the condition, because statistically you'd expect them to be about the same. Quality score based on linkage disequilibrium. We'll talk about this when we look at neurodegenerative diseases, but you can impute genotypes for SNPs that you don't directly genotype based on linkage disequilibrium. We don't need to get into that right now. Probably the most important column is odds ratio. This describes your odds of having of being a case given your genotype on this allele. So this is really just kind of a function of looking at the frequency of that allele in the cases and the controls. And so, for every copy of the alternate allele you have for this, your odds of being a case go down a little bit, maybe 2%. For every copy of the alternate allele you have on this one, your odds of being a case go up a little bit, and so on and so forth. So you can see there's this odds ratio value given for everything. Most of them hover around 1, because for most SNPs, it doesn't matter whether you have, like, which allele you have. It won't affect whether you're a case or control, because even in the condition of ASD, they only found 5 of these that actually were significant. So only 5 SNPs in the whole genome, according to these authors, were significant enough to meaningfully change your odds of having ASD or not. And then you get standard errors. That's not too important. P-value is the statistical significance of this. None of these are significant. I only picked the first 24 SNPs off a list. So anything with a p-value of less than 0.05 would be significant. But the cutoff for genome-wide significance is much, much lower than that. It's 5 times 10 to the negative 8 or something like that. So there's a much lower cutoff. And then you have the number of cases and controls. So we've got, in this particular example, for this SNP, you've genotyped 33,000 plus cases and 43,000 plus controls. And the number can vary between SNPs, because sometimes the quality control of the genotyping is low enough that it's not included. So you don't have totally even numbers for every example. So now that you have that, what you can imagine is taking one set of GWAS summary data for, like, let's say, how about schizophrenia, and then taking the same SNP data, or taking the same kind of data for bipolar disorder, and then just lining the SNPs up together, 1-to-1, and then plotting them against each other, plotting the odds ratios against each other. Okay, you can do this because all these studies tend to focus on the same SNPs. And what you will see in some cases is a genetic correlation. So what I'm showing here is an example that I made for you to look at. It's kind of a simplified version of things, but I've taken the odds ratio for a bunch of bipolar disorder associated genes. You can see that, alleles. So for example, this point right here, if you have the alternate allele on that particular SNP, your odds of being diagnosed with bipolar disorder are almost, let's say 1.4. So they're about 40% higher than if you didn't have it. Okay, that's a pretty high effect size allele. Now, if you look at that very same SNP and ask how it relates to schizophrenia, what you will find is that if you happen to have that same allele, your odds of being diagnosed with schizophrenia are also a good bit higher. They're closer to 1.3 or so. So in both cases, having this alternate, having this particular allele raises your odds of having either bipolar or schizophrenia. So this is a positive genetic correlation. It turns out that when you look at many of the bipolar associated genes, or SNPs I should say, they have the same relationship with bipolar as they do with schizophrenia. If you have the alternate, or sort of the bad version of that allele, or the bad version of that SNP, it increases your odds of either schizophrenia or bipolar disorder. And the same would be true for the ones that protect you. So say for example, let's look at pretend we have one right here. If you have the the minor allele here, then your odds of being diagnosed with bipolar disorder are 0.75. So they go down by 25%. And at the same time, your odds of being diagnosed with schizophrenia also go down 25%. So the same relationship obtains for both disorders at this particular SNP. And the same would be true for any SNPs that are kind of close to this line of best fit, this regression line. So this is a positive correlation. Now, there are some SNPs where there's not a positive correlation. Here are ones where if you have this particular allele, your odds of bipolar disorder increase by about 25%, but your odds of schizophrenia actually decrease by, maybe 20 some odd percent. So there are instances where the correlation is in the reverse direction, but you can see that of this example at least, most of the SNPs are positively correlated, and that only a handful of them have this negative correlation running in the opposite direction. So if you look at this, you can see that the genetic architecture of bipolar disorder and schizophrenia it's actually fairly similar. A lot of the same genes, or a lot of the same SNPs, have similar effects on both phenotypes. Now, this isn't always how it works. This isn't just like you can't just throw any two data sets together and find a positive correlation. If you take things that have nothing to do with each other, you'll find no correlation. So let's take, as an example, we'll take schizophrenia and we'll plot the odds ratio of that on the x-axis. Here we've scaled it using the Z transform, so the numbers are going to be different. You kind of have to do that to make it comparable to the height, which is a beta value. So these are standardized values, unlike here, which are raw odds ratios. So they're a little more tough to interpret. But anyway, the alleles that increase your height have no real relationship consistently with schizophrenia. Now, some of them do, like all the ones in this quadrant, they make you taller, but they also raise your odds of schizophrenia. But all the ones in this quadrant make you shorter, but increase your odds of schizophrenia. All the ones in this quadrant make you shorter, but decrease your odds of schizophrenia. And all the ones on this quadrant make you taller and decrease your odds of schizophrenia. So each of the four possible relationships is reflected here, such that when you plot a regression line, it basically is flat. And if you remember from your statistics class, if your regression line is flat, there's no correlation. So there's no relationship there. The genes that contribute to height have no relationship at all with the genes that contribute to schizophrenia. And that makes sense because height and brain development are two separate things, largely. But brain development as manifested in schizophrenia or bipolar disorder, that's a kind of similar thing. Those are very similar conditions in terms of their symptoms, in terms of their the way that you would maybe treat them, and genetically speaking, they're also very similar. So that is a genetic correlation, and I think that's a very, very important point to make that I hope you are all, I hope that you all understand and kind of cherish, because I think it's one of the more, I really think it's one of the more interesting things, because it shows us connections between phenotypes and conditions that we would never, or that we never really would have thought about outside of the world of genomics. Sorry, I just choked on some water there. So let's look at genetic correlation on a large scale. Now that you see it's possible to combine these GWAS data sets and identify correlations between traits that you may or may not have thought have any relationship, well, there's a lot of GWAS data out there, folks, and you can actually correlate pretty much everything. So why not just make big correlation maps of all the possible things and see if anything interesting comes up? And that's precisely what they did here in 2015. So this is Bullock-Sullivan et al., published in Nature Genetics. So let's just kick back and enjoy this plot for a little bit. This is a correlogram. A correlogram expresses the results from a large number of pairwise correlations. So on the X and Y axes of this figure, you have all the different traits that were studied: smoking, BMI, childhood obesity, fasting glucose, type 2 diabetes, etc. Down here you kind of have like anorexia, depression, bipolar disorder, schizophrenia, years of education, autism spectrum, that kind of thing. So you have all these things together, and then if you follow this along, you can so if you follow from one a line, or if you follow from one row to one column, you can identify, or you can see the correlation that's been calculated between the genetics of these things. So naturally, and then the strength of the correlation is given by the color scale. So dark blue means a correlation coefficient of 1 that is perfectly correlated. That's as positive a correlation as you can possibly have. And dark red is a perfect negative correlation. And then intermediate levels express intermediate levels of correlation. Now, by the nature of this plot, every trait is perfectly correlated with itself. So if you start up here, ever smoked, the genetics for that are perfectly correlated with ever smoked because you're talking about the same thing. And the same thing is perfectly correlated with itself. It's sort of a mathematical tautology. That's not too interesting. But let's look at some other correlations. Let's take BMI, for example, body mass index. What we can see let's trace over here to childhood obesity. Body mass index is positively correlated with childhood obesity. Not surprising, really, because your body weight as a child has a fairly strong relationship with your adult weight. BMI is also positively correlated with things like coronary artery disease, type 2 diabetes, triglyceride levels. These are all aspects of metabolic health that have a connection with BMI on the sort of the physiological level. The more fat you have, and BMI is sort of a measure of fat, although it's far from a perfect one. The higher your levels of triglycerides tend to be, and the higher your levels of other kinds of, your higher rate of type 2 diabetes and stuff. However, what this is showing is that in addition to that physiological correlation rooted in hormones and lipids and whatnot, there's also a genetic correlation, meaning that the same genes that raise your odds of having a higher BMI independently raise your odds of having type 2 diabetes. And then that's all well and good. Let's look at some other ones. It's negatively correlated with HDL levels and age of menarche. Age of menarche is when you have your first period, if you happen to be somebody who has periods. It's also negatively correlated with height, and it's also negatively correlated with years of education and anorexia. So that's kind of an interesting one, actually. The genes that contribute to your diagnosing, or you're being diagnosed with anorexia nervosa, those genes are kind of the opposite of the ones that lead you to have an increased BMI. Perhaps not surprising. Anyway, you can go down this list and explore all these, and you'll see these clusters of correlations where, for example, the genes that cause Crohn's disease are very similar to the ones that cause ulcerative colitis. The genes that cause or that have to do with anthropomorphic characteristics like height and infant head circumference, these are all positively correlated with one another. In height, infant's head circumference, birth length, and birth weight are all correlated. And, that's great. We're all happy to hear that. We're really here for the mental illnesses, of course. So we can look down here, and what you will see is that the genes that are associated with depression, bipolar, and schizophrenia form this positively correlated cluster. So it turns out that these three conditions, which you might think don't have a huge amount to do with each other, are actually fairly well correlated genetically. They're all the genes that are affected in each of the three cases have to do with the brain. They're neurotransmitter signaling things, they're receptors, they're growth factors, etc., etc. There is a fairly strong genetic correlation between all of these. There's also a correlation between these and years of education and anorexia. These are, of course, all sort of aspects of brain development that can be measured genetically. So very interesting kinds of stuff that you can look at if you look at genetic correlation on a large scale, really pointing to a common underlying architecture of these conditions. So we got a question in the chat. So Natalie asks if I'm not sure if you mentioned this already, but what do the plots with a circle or square inside of them mean? Right. So in this figure, and I don't think I really mentioned that very clearly, although I don't really know what the square or the circle. Oh well, yeah. So the asterisk here just means that the relationship in that correlation is statistically significant. So correlation is a number that you can measure for anything, but if it's not a statistically significant correlation, then it doesn't pass the kind of threshold for being reliable in a statistical sense. So the ones with stars or these little asterisks, these are the ones you want to focus on more. And then the size of the square represents the statistical significance as well. The star represents significant at a very stringent level where like you're setting the bar really high, and the size of the box represents a 1% false discovery rate. So if the box is smaller than fully filling the square, then it's really not a significant correlation, and even though it might be there, it's not worth paying attention to. If the box fills the square, that means it's it's sort of approaching being statistically significant, but it's sort of on the borderline of like, maybe you don't want to pay too much attention to this, but it might be interesting. And if the box fills the square and has a star, that means it's really significant at a very stringent level, and it passes the kind of the threshold for paying attention. So hopefully that clarifies it. Anyway, when you're playing along at home or studying, to just take a look at this figure. I think it's really quite fun to kind of just explore these correlograms and look at the different correlations and relationships you can find between conditions. I find that to be a very enlightening topic, I guess, to consider that. All of these conditions that we're curious about, mental illnesses in particular, all have some kind of underlying genetic structure to them. Because, we're all very familiar with the fact that the means of diagnosing and categorizing illnesses are not perfect. The DSM changes every once in a while because they think of new ways to define disorders, and disorders change around. One year you might have, ASD, and the next year you pass below the cutoff line for it, et cetera, et cetera. Some diseases completely fall off the map. Some things aren't even diseases anymore. They were just incorrectly classified. So it's just fun to look at the genetics because these are at least a little bit more objective, you would think, although I guess they still depend on people having a diagnosis before they go into the study, so they're not perfect by any stretch, but it just kind of points to something that you wouldn't be able to find otherwise. So I'm bringing this all up because we have to get back to ASD, and we were kind of, I think, maybe a little disappointed with the amount of discoveries we made using GWAS. 5 SNPs, that's not very good. 5 is not going to help anybody, really. But you can kind of gather strength from other traits. You may not have enough ASD. You may not have enough people with ASD to ever really find all of the variants that are linked to it. However, if you do genetic correlation analyses, you might be able to get hints about variants that are important based on their correlation. So, for example, there's a positive correlation between ASD genes and IQ, educational attainment, whether or not you went to university, self-reported tiredness, neuroticism, schizophrenia, major depression, depressive symptoms, and ADHD. So a whole host of things are positively correlated. Some of these up here, I would say, are probably things that are okay. Maybe not tiredness, you wouldn't necessarily want that, but educational attainment, IQ, and college, those are all things that we would consider to be fairly positive in society. Self-reported tiredness, not so much. Neuroticism is just a dimension of personality, but I don't think anyone is out there, begging to become more neurotic. And then you have a number of mental illnesses. So it's a little difficult to kind of interpret this, but what it seems to suggest is that the genes that are related to ASD are brain-related genes. So you may not be able to prove a connection between some of the genes, between some SNPs and ASD, but if you say that, IQ is positively correlated, maybe you can look at the genes or the SNPs that are linked to IQ and start to build out on those without ever fully proving the relationship with ASD. You might just study them in particular and see if you can manipulate them in rodents and produce ASD-like phenotypes. It kind of gives you a hint of where to look, even though the significance of the direct ASD versus case versus control comparison isn't powerful enough. It's worth also noting that it's negatively correlated with subjective well-being and with chronotype. So I don't know why that would be. Chronotype is kind of weird. Chronotype refers to sort of whether you're a morning person or a night person, whether you like getting up early. I personally have discovered, this can change, I think, because I didn't really consider myself to be a morning person, but now in my my later years, I've certainly become a morning person. Typically, I wake up at around 5 a.m. to start working, and so the day's almost, the day's already well and truly started for me by the time I get to teaching this class. But anyway, that is an aspect of your phenotype that's apparently negatively correlated with autism, but I don't really know what the measurement here even is. But anyway, this gives you a starting point for understanding ASD, even though the genetics of ASD itself are still mysterious. You know that it has something to do with these aspects of brain function and brain development. These are all brain-related traits. So it gives you a starting point you can kind of work with. But I think you'd be right to say that we're not totally satisfied with where we're at in terms of our understanding. The best GWAS we have has a very limited success overall. 5 SNPs is not going to get you, it's not going to help you understand ASD, because it doesn't even, those 5 SNPs don't guarantee you have it. They're just associated with it. Now, you might need larger sample sizes, but it's tough to get that. How are you going to find 50,000 people with ASD? and have enough resources to phenotype them and figure out what the nature of their condition is and control for all the other factors. It's also possible that ASD depends heavily on combinations of rare variants. GWAS studies don't typically find rare variants because they're designed to look at entire populations and find associations between relatively common variants and a condition. Rare variants that are visible in less than 1% of the population are not even examined by GWAS studies for the most part. So those could be hidden beneath the surface and contribute to a huge amount of ASD causality without ever being detected by the GWAS studies themselves. And what's more, there could be features that influence ASD that you would not be able to pick up using a normal GWAS screen. Remember that GWAS studies can only really look at SNPs. They can sort of maybe a little bit address copy number variations and indels, but for the most part, they're really just made to look at SNPs. And they're made to look at common SNPs that we all already know about. They can't discover new SNPs. They can't discover major rearrangements of the genome. And they can't discover de novo mutations because SNPs or genotyping arrays are pre-designed with SNPs that we already know about. De novo mutations are new mutations that are unique to you as an individual or to your family line, and they're not part of the overall, they're not part of the known human genome, I guess. Now, these variations are oftentimes random, and they are oftentimes unique to the individual, so they're not even inherited from parents. And because they're new, they're not typically identified by GWAS screens. They, documented SNPs by definition can't be de novo. They would have been de novo at one time or another, but they're not de novo now because we know to look for them, so they're not new. And so it's been estimated that de novo mutations could account for up to 30% of simplex ASD cases. A simplex case is when you have a family where one sibling has ASD, but the other siblings and the other parents aren't diagnosed. So it's a case where it's a brand new case of ASD that's not been running in the family. You can't point to a parent or a grandparent or somebody else who has it. And ISOF and company back in 2014 used exome sequencing. So this is when you use genome sequencing of just the exons of the DNA. And that is sequencing is a method that will detect de novo mutations because you're sequencing it. You haven't planned ahead of time what you're looking for. You're just sequencing out the genome and asking what it's got. That's why the title of the slide is "Okay, genome, what else have you got?" Because the SNPs aren't cutting it. So what they find is that the rate of mutation events in ASD children, the red bars are ASD, are oftentimes significantly higher than those in their non-ASD siblings. These are simplex families, so the ASD case is kind of unique. The p-value for this comparison, or the statistical significance, is given here. God only knows why they picked red and green as the colors. It's very disrespectful to people such as myself who are colorblind. You really have to think about how, I don't know, it's like having a building where it only has stairs inside of it or something. It's just kind of silly. But why not do black and white? Why not do, yellow and blue or like any other kind of color other than red and green? But anyway, so what you'll see is that the number of de novo mutation events is higher in the ASD case in a variety of contexts. So there's more substitutions. Those are cases where the amino acid is swapped out. They're not more synonymous ones. Synonymous ones is when you have a mutation to a codon that changes the codon but not the amino acid it codes for. So those are typically pretty harmless. Missense ones change the meaning of the amino acid. Those are significantly higher in ASD patients, as are nonsense and RNA splice site mutations. Indels are more common in ASD patients, as well as frame shifts. Frame shifts are really nasty ones. That's when you actually get an insertion of a nucleotide in an exon that shifts the codons forward by one position, thus totally ruining all the downstream genetic information. So they are, there's a whole range of de novo mutations that have much higher rates in individuals with ASD than in their neurotypical siblings. Let me just go back to the chat for a minute here. So Nathalie asks, since GWAS can only highlight SNPs, it would be redundant to do another GWAS on ASD, since we'd probably see the same 5 SNPs that were found before. That's probably true. The one I would make, if you were to pick another, if you were to go to a different population, for example, look in a different continent, look in Africa, or look in Asia, or look in the Pacific Islands, or look pretty much anywhere other than Europeans, you would probably find new variants that we didn't find on the past one, maybe, or maybe you'd find the same ones over again. So there would be a benefit to doing more GWAS studies on different population groups where you might find more novelty. And, if you added another 20,000 people to the study, if you found another 20,000 people with ASD who haven't been looked at before, you probably would get some new hits that you hadn't that you hadn't seen before. But it starts to get a little bit pointless because, do you really want to go and do this again? It's like going to the moon a second time, in a sense. Like, we've been to the moon. We don't need, we kind of get it. We see what's up there. Is it really worth all the expense and time to go back again just to get some more moon rocks? Maybe you would find a new kind of moon rock, but you probably know what you're going to find. So yeah, I think that you'd want to do that in different population groups, but it would be a hard sell for people because you'd really need to increase, you'd need to make a leap in technology to make it worth going back to the same population and looking at them all over again. As a punchline, what you'd really want to do is move past SNP genotyping and do whole genome sequencing. Then you might have quite a lot of power, but that's still a little too expensive for everybody. Okay, so I'm just going to take a quick break to get some more water, and I'll be back in 1 minute, folks. And we're back. So the last thing I wanted to point out, I guess, was that in your studies, you don't have to worry about what these are, like what an LGD is or what a frame shift. You should know what a frame shift is, but I'm not going to quiz you on that. Just know that on the basis of exome sequencing, which is a method that sequences the exons of genes, the coding regions, the authors found that ASD cases had higher rates of mutation compared to their siblings who didn't have ASD. So you have the families are the same, you have the basic kind of genetic background is the same, but the rate of mutations is much higher in the ASD case. And this would suggest that ASD in part is due to de novo mutations, which of course would be impossible to detect using GWAS studies. Let's look at another question here, because if we have mutations, we have to ask where they're coming from. Typically when there's a de novo mutation, it's happening in either mom or dad's germ cell, either in the sperm or the egg during meiosis at some point, develops a mutation as the DNA is being copied. This is very possible. It's pretty normal for that to happen, actually. This is part of the place, it's part of where human genetic variation comes from. But you would want to know if you have a de novo mutation that you've linked to ASD, you'd like to know whether it comes from mom or dad. Now, how can you do this? It's de novo, so you can't see if the mutation is there in dad or in mom. You can't sequence mom or dad's DNA and find it there because it's new. So how do you find it? Or how do you identify it? The answer is with haplotype phasing. It's possible in some cases. So let's say you have a sequence of DNA that you've read out on the sequencer. And so it's going from, 5' to 3', so it's kind of going in this direction. I'll draw a little arrow on the end of it. Let's say on that sequence read, you've got a mutation. We'll draw a little circle here. So this is your de novo mutation. Now, how do you figure out whether this mutation came from the sperm or the egg, whether it came from mom's DNA or dad's DNA? Well, you don't know, you can't go back and measure either of the parents and find it there, but what you can do is look elsewhere on the DNA to see if there's another SNP that you can genotype and then link that back to mom and dad. So let's say upstream of this de novo mutation, you also have a SNP that you know is only present in dad, let's say. So if you genotype that SNP and you know it's on the same stretch of DNA as the new mutation, you can say with a good deal of confidence that this mutation must have happened in the sperm and have come from dad. That's called phasing. Phasing is when you use information on the sequencing reads to identify which parent they came from or which haplotype that they are located on. And this is precisely what has been done in this paper. Now, maybe you've heard before that the rate of ASD goes up with paternal age. The older dad is, the higher the rate of ASD is in their children. And there have been a lot of thoughts about why this could happen. It could be, for example, that mutations build up. That's what we're sort of suggesting here. It could also be that dads who have kind of autistic spectrum traits, who maybe don't socialize quite as naturally, take longer to find a partner and achieve kind of romantic intimacy needed to have to procreate. Maybe it just takes them longer, so you have this kind of effect where dads who are prone to ASD tend to just naturally take longer to reproduce. That's also conceivable. Both could be true at the same time. But anyway, this should say paternal. Another mistake. See, I love finding mistakes that I've written. De novo mutations that they found occur 3 times more often on the dad's DNA than they do on the mom's DNA. Now, this is, kind of expected in a way, because dads produce vastly more sperm than moms produce of eggs. Eggs are, you have a fixed number that are produced during your embryonic life, and that's as many as you get, whereas males are continually churning out sperm all the time, and you would think any factory that makes millions of something is going to make a lot more mistakes. It's like the difference in quality between a piece of furniture you get at IKEA and something you would get from like the Emporium or some type of fine furniture place where everything's made by artisans. Anyway, so the authors confirmed that the rate of mutations is positively correlated. So the mutations 3 times more often come from dad's DNA than they do from mom. So 3 times more often they're coming from dad. And the rate of mutation is correlated with paternal age at the time of conception. So here we have the plot that shows this. The y-axis of this plot is the number of exomic mutations per child. So the higher means the higher rates of mutations. They're looking here at substitutions, which is a particular type of mutation. And then on the x-axis, you have dad's age at childbirth. So some dads could be as old as 50, other dads could be in their early 20s. And what you will see is that there is this nice positive correlation between the age of dad and the number of mutations that you see in their autistic child. This is pretty clear evidence, I would say, that the observed effect of paternal age has to do with the rate of mutation in the sperm. Let's get into another topic now. We're asking what else the genome has for us. We feel like GWAS studies haven't really delivered. We need more. Well, there is another kind of thing that GWAS studies can't find, and these are tandem repeats. Now, as I said, repeats are coming back for this class. We've looked at them in the context of Fragile X, and we've looked at them in the context of Huntington's, and now we're going to see what they have to do with autism. So I've given here, now that we're kind of zooming out and talking about repeats in a more generalized way, I'll give you a hierarchy of repeat types. Throughout the genome, between 50 to 75% of it in the human is repetitive, depending on how you define it. So maybe only a quarter of the genome is actually kind of informative, meaningful genes. The rest of it is kind of repetitive stuff of various sorts. Repeats can be categorized either into tandem repeats or dispersed repeats. The dispersed repeats constitute retrotransposons, which are viral elements left over in the genome from lines and signs and these kinds of things, like viral elements that have been inserted into our DNA, as well as DNA transposons. We might talk about those later, but not right now. And then within the tandem repeat category, we have satellites, mini satellites, and microsatellites. Microsatellites we've talked about before. Those are markers used in repeat fragment length polymorphism analysis. So tandem repeats are regions where a single short sequence, 3 or 4 nucleotides maybe, is repeated many, many times. And as I've said, if you want to know what this is in practice, just go back to our talks on Fragile X and Huntington's disease, because those are both caused by expansions of tandem repeats. Tandem repeats are a broader category. Those are trinucleotide repeats, as I call them there, because there's 3 nucleotides. But trinucleotide repeats are just one example of the broader category of tandem repeats. Microsatellites are short tandem repeats as well. We've already heard about microsatellites. So now I'm kind of bringing this all together. What we're talking about when we talk about all these kinds of repeats are basically a type of tandem repeat that is a broadly represented motif throughout the genome. Now, importantly, the number of repeats is extremely variable between individuals. You might have 10 repeats, you might have 20 or 30 or 40. It's extremely variable, much more variable and much more polymorphic, I should point out, than SNPs themselves. With SNPs, for any given location, you can either have the regular allele, you can be heterozygous, or you can have the alternate allele. So there's 3 possibilities, basically. That's not a lot of variation, three possible choices. With tandem repeats, there's a huge range for polymorphism, because you might have 1 copy, or you might have 10, or you might have 100. There's a huge range across which you can vary, much more than the 3 positions you can vary for a typical SNP. So tandem repeats are very good for genotyping people. They're used in DNA fingerprinting, which would have been an older technique for forensic analysis of DNA. It's also used in linkage studies, as we've talked about. So a very sort of central theme in the structure of the genome. What does this have to do with ASD, though? Well, actually, before I get into this, let me bring up that tandem repeats are difficult to sequence. So we're kind of asking, why aren't tandem repeats seen in microarray studies, GWAS studies? You can't measure them in GWAS studies because they're repetitive. If you just take a piece of the repeat, you have no idea where it came from or how many copies the person has or where in the genome it came from. For the same reason, they're difficult to determine even using DNA sequencing. So even using the highest level of technology we have, it's very tough to tell how many repeats somebody has across their genome. You might be able to, you can take one area and measure the repeat length using like gel electrophoresis or southern blotting or something, but that's way too time consuming to do on the scale that we would need it. If you're trying to make a point that the repeat length has something to do with ASD or any kind of other complicated condition, you have to determine it using genome-wide sequencing. But they're very difficult to sequence, and the reason is pretty simple. Let's say you've got a sequencing read. Let's say this yellow highlighted region is a repetitive region. You can just see it's CAG, CAG, CAG endlessly. If you have sequencing reads that cover flanking sequence, like these blue arrows here are reads that cover flanking sequence, you can kind of anchor them. So here this read covers GAT, CAT. That's a unique sequence, and that can be mapped to the genome using BLAT, for example. And then the rest of it hangs out into this repeating region. The same would be true for this sequence. It's flanking, or it covers some of the flanking sequence, so you know where it's located in the genome, because this flanking sequence would be unique. And then it hangs over into the repetitive area, so for a certain amount, you can cover the repetitive area. But any of these red arrows are impossible to place, because this red arrow here, this read, it could have come from here, but it could also have come from here, or it could have also come from here, or it could have also come from here. You have no idea how to place them because they're in the middle of a repetitive area. So they can't be mapped back to the genome because you don't have a specific enough location for them to be mapped to because it's a repetitive area. So it's very, very difficult to sequence these things, and it's impossible to do it by microarray, the kind of method used in GWAS studies, but it's also very tough to do by sequencing. So if you're trying to estimate the length of repeats, you would have to have reads that are long enough to cover some of the flanking sequence and then reach deep enough into the repetitive region to measure how long it is. Maybe even be able to be long enough to reach the whole way across. That's the challenge, and it's very, very difficult to design studies that can do this. So what I'm saying is that tandem repeat length, genome-wide, is kind of an unexplored frontier of variation. We already know it varies a lot. I mean, people... the DNA fingerprinting studies and the linkage studies show that repeat length varies hugely between people. But that knowledge hasn't yet been applied to genome-wide studies of complicated traits like ASD, or at least it hasn't. It's just beginning to be. That's the challenge. It's very difficult to deal with on a technical level. So what Mitra et al. Found just this year, in fact, and I'm giving you the latest stuff. I'm trying to keep keep things as up to date as possible in this class. You'll never hear this, this is the only place you'll hear such fresh, tasty information. But anyway, they did a study where they had developed, what amounts to a computational algorithm for processing whole genome sequencing data, WGS is whole genome sequencing, to genotype tandem repeat length. So they found a solution, or they developed a system for using genome sequencing, next-gen sequencing, to measure repeat length across the genome. And then they did a study of simplex families. Remember, 1,600 some odd simplex families. Simplex families are where one kid has ASD, but the other kids don't. And what they found is that the rate of tandem repeat mutations was significantly higher in probands. Probands are the people who are actually cases than their control siblings. And these mutations typically involve expansion or contraction of the repeat sequence by at least 1 unit. So this is a histogram showing the number of tandem repeat mutations per child. The probands are the red bars and the siblings are the blue bars. And this is actually not made very well, but what you can see is that there are more of the probands in the higher number of mutation bins. So like if you were to draw these as kind of density plots, you'd see the siblings are here and the probands are here. So there's a shift. The probands have higher rates of tandem repeat mutations than the siblings. And this also correlates with the age of the father at birth. So here on the x-axis of this scatter plot, we have age of dad. And then on the y-axis, we have the number of tandem repeat mutations seen in their child who has autism. And there is a positive correlation there. It's not a particularly strong one. The correlation coefficient is 0.19, which is not, earth-shattering or anything. But this is very significant, mainly just because there's a lot of people in the study. But anyway, there's a positive correlation here. So the rate of mutation is correlated with dad's age. And what's more, thanks to phasing, 74% of the tandem repeat mutations were phased to the paternal genome. That means that 3/4 approximately of the mutations came from dad, and those correlate with age. So you see we have this similar kind of story. The rate of mutations in sperm goes up as dad ages, and a variety of mutations, whether they're de novo mutations in exons or whether they're tandem repeat mutations throughout the genome, all seem to contribute to ASD risk. And both of those, I should say, impossible to find using microarrays. You cannot do phasing on a microarray. You have to have sequencing data to do phasing. So pretty interesting stuff. Now, if you look at where these tandem repeat mutations are happening, what you'll find is that they fit into a variety of genomic annotations. So overall, here's the bar graph showing rate of mutations in siblings, the blue bars, and in probands, people diagnosed with ASD, significantly higher throughout the genome in all contexts. There's no difference in 5' untranslated regions or 3' untranslated regions. There's a significant difference in the fetal brain promoters. So these are regions of the DNA that control the expression of genes during fetal brain development, okay? Now, this is quite telling because we think that ASD is a neurodevelopmental disorder. So if the mutations are all concentrated in parts of the genome that control the expression of genes involved in fetal brain development, you have this perfect story where the mutations affect brain development during young life, and then of course if your brain doesn't develop the typical way when you're very, very young, it's permanently going to be different. It will begin to manifest as the world becomes more and more challenging for you and as social situations demand it. But the actual injury or the actual difference is established early in life during fetal brain development. So there are substantially more of the probands here. And in fact, this is the area that had the strongest relationship, at least on the basis of odds ratio. So RR means relative risk. So for every the more of these tandem repeat mutations you had in the fetal brain promoters, your risk of ASD went up by about 20% or 1.2-fold. So this is a fairly, a reasonably strong effect. It's far from explaining all of ASD, but it certainly gets us started and shows us what else the genome has to offer. And then finally, if you look at the genes where ASD-linked tandem repeat mutations are found, using an existing database of genes that are expressed in various brain regions, what you will find is that the genes that have these mutations are typically ones that are expressed in the brain. So that's what they're sort of showing here. These are data from human brain studies of gene expression in various areas. So Amy is amygdala, dorsolateral prefrontal cortex, hippocampus, I don't know what's inferior parietal cortex, inferior temporal cortex, etc. Going down the line, looking at all these different brain regions. And then the level of mutations in the siblings is given by the blue lines, and the level of mutations given by the probands is, the mutations given by the probands is in the red lines. And so the genes where you have, that's kind of a little bit of a confusing plot. My apologies for choosing it. Well, really the author should be blamed on having a weird plot. But basically, genes that have mutations in probands, because probands genes that have mutations in siblings, the people who don't have ASD, the expression level on average of those genes is lower in these brain regions than the genes that have mutations in the probands. That's kind of a confusing point, to be completely honest. Like, siblings have mutations, but the genes where those mutations happen don't tend to be brain genes. They tend to be other genes. The ASD probands have mutations, and those mutations happen to be mostly in brain genes. I think that's how you would summarize it. This is really not a great looking figure. But anyway, I guess that's enough to say that, like, this is clearly something where the mutations are concentrated in such a way as to affect brain development. And just to be crystal clear here, maybe I didn't exactly spell this out with a diagram, but what I mean by a tandem repeat mutation is either an expansion or contraction of the number of repeats. So think about, it's just the very exact same as Fragile X or Huntington's. In, say Fragile X, you might have 20 repeats, but if it happens to expand to, say, 50, your odds of the condition go up because the repeat has expanded considerably. Now, that's a repeat expansion in a known area that we can measure on its own specifically. Everywhere else in the genome, the repeats might be expanding or contracting. They just happen to expand and contract in areas where they're not causing like such a harmful effect. So repeat expansion and contraction happens all over the place in the genome. It's just the fact that most of the places where it happens, it's not terribly harmful. In the case of Huntington's and Fragile X, those two repeats happen to be located in a place where if they expand, it's very, very bad for you. But in most places, it doesn't have such a strong effect. But it does have some effect because expansion and contraction of repeats in brain-related genes does seem to increase your odds of developing ASD. So hopefully that makes sense. Anyway, my friends, that's all I have for you all today. I guess just to summarize, what I'm trying to show you here, following my master plan for teaching you all that I know about genetics and genomics in the brain, is to show you that for conditions like ASD that are clearly genetic to a certain extent, oftentimes the normal GWAS methods don't give us enough to understand. They don't give us enough to go on. So we looked at the GWAS study. We're not too pleased with it. Then we're asking the genome, what else does it have? What else is in there that could be different between people and could potentially contribute to ASD that would be not identified by the normal GWAS method? And what we found are things like tandem repeat expansion, which can only be detected by sequencing, and we found de novo mutations, which can only be detected by sequencing. The perspective then, or the overall kind of take-home message, is that there's a lot more to be found in the genome, probably for every condition, that can't be found yet because we do not yet have the infrastructure or the capacity to do whole genome sequencing on every person we study. If you were to do a study where you took in 20,000 ASD patients and 20,000 controls and did whole genome sequencing on all of them, you would probably find a huge amount of interesting stuff, but we kind of know it's there, but we don't yet have the resources to do it. So this here is a model case of the promise that is held by technology to potentially explain more than just ASD, but other cases where we also, other conditions where we don't have enough data. So that's it for now. We got the exam coming up on, or the quiz rather, just a quiz, coming up on Monday. I kind of realized I think I said everything I wanted to say about that on the last class. So I gave you some hints or some pointers of things to study. Obviously, like, your kind of goal as students would be to like try to make connections across lectures. That's what I will try to focus on again for this. But here's an obvious one. Tandem repeats, now we know they are involved in 3 separate conditions. How are they involved? Compare and contrast. That's a very simple kind of question that could be asked. It asks you to kind of integrate knowledge from a variety of topics and combine it all into a narrative that sort of conveys your understanding of it. So that's something to think about, and any more, if anything else comes to mind, I'll put it in the exam as well. Anyway, so that's it for that. So stay tuned for that going live. We'll have your marks ready for the assignments probably by this time next week, and we'll be able to get the marks up there. The 3rd assignment will be released soon. I have to record the tutorial video for it and have the TAs try it out, but that should be released in the next week or 2, and you'll be able to get to that. And yeah, office hours is tomorrow, so please stop by if you have any questions. And until then, my friends, we'll see you next time.